Is Gene Editing of Human Embryos Coming Soon by Dasha Bond. What is gene editing? Genome or gene editing is when scientists can change a living organism's DNA with the use of technology. The technology allows scientists to add, remove, and alter parts of the genome or gene. Technology used in gene editing. We have transcription activator like effector nucleases or TALENs or talents, which are restriction enzymes that can be engineered to cut specific sequences of DNA. They are made by fusing a tau effector DNA binding domain to a DNA cleavage domain, a nucleus which cuts DNA strands. So I guess in this way it acts as scissors. Then we have zinc finger nucleases, ZFNs, which are a class of engineered DNA binding proteins that facilitate targeted editing of the genome by creating double-strand breaks in DNA at user-specified locations. To me, this sounds like it acts as a glue. Some other technologies used include CRISP and CRISPR. These are adapted from naturally occurring genome editing systems within bacteria. The bacteria captures snippets of the DNA from invading viruses and uses them to create DNA segments known as arrays. These arrays allow the bacteria to remember the virus. If that virus attacks again, the bacteria produces RNA segments from those arrays that targets the virus. That bacteria then uses Cas9 or a similar enzyme to cut the DNA apart, which resembles scissors. We have homing endonuclease or meganucleases that has become a valuable tool for the study of genomes and genome engineering over the past 15 years. Meganucleases are molecular DNA scissors that can be used to replace, eliminate, or modify sequences in a highly targeted way. What is gene editing currently used for? Well, genome editing is currently used to edit human genomes, but only somatic cells. And these changes are not done to sperm and egg cells. This means that the changes are not passed down from generation to generation. Editing somatic cells targets tissue that aids in gene therapy and treating certain diseases. Gene editing is also used in crops. This results in crops yielding more food, plants, and things like that over time. Here we have pros and cons of gene editing. Some pros of gene editing include crops yielding more food that fights hunger. We have gene therapy, it treats diseases, and it may be able to cure hereditary diseases in the future. Some cons, just like anything else, include negative effects on human health. That's more linked to editing the genes of plants that produce those high-yielding crops. There are also a lot of unknown effects of genes that weren't intended on being modified. And then science's biggest fear, which is designer humans. There's currently a fight on the ban of human embryos being gene edited due to a Chinese biophysicist named Jiankui He. In November of 2018, he created twins by editing them as embryos so that they would be immune to HIV. This definitely sparked an uproar due to the experiment being extremely unethical due to the unknown complications and effects it would have on the twins. This also produced debates all over the world. Scientists are now calling for nations to make laws that prohibit gene editing human germline cells and prevent studies surrounding this topic until they know for sure that it's safe to do so. In my opinion, based on all the information, gene editing of human embryos is not coming anytime soon. I say that because scientists are currently trying to prohibit gene editing germline cells, and the technologies used to edit genes for other purposes is great, but it presents great risk when it comes to editing human embryos.